Hello everyone, I am Hela Chikh-Rouhou from Tunisia, working in the Regional Research Center on Horticulture and Organic Agriculture. I am working on the valorization and preservation of cucurbits local landraces and their exploitation for sustainable agriculture. The crops I'm working on are watermelon, pumpkins, and bottle gourd. And currently, I am managing a melon breeding program for biotic stress resistance using phenotyping and molecular approach. And the talk of today gonna focus on the identification of Tunisian melon landraces resistant to biotic stress for their use in breeding programs. I will give you an overview of the things done in our research center and how to achieve our goals. Melon is a vegetable crop of high nutritional value and economic importance in Tunisia and in the Mediterranean regions. And because of its strategic position and the diversity of ecosystems and climatic conditions, Tunisia is considered one of the richest Mediterranean countries in terms of plant genetic resources, including landraces and wild relatives. Why landraces? Landraces are genetically diverse and well adapted to local environmental conditions. They are considered as an important reservoir of useful genes that could be exploited in crop breeding programs. And here in Tunisia, there is a lack of information regarding this germplasm, and also little attention has been paid to its conservation. So the evaluation of these local landraces to biotic stress is promising for the identification of resistance genes, but also to ensure the valorization of this germplasm. Melon crop is affected by several pathogens and pests, and Fusarium wilt, powdery mildew, and Aphid stand out among them. Fusarium wilt is a soil-borne disease caused by Fusarium oxysporum, form special melonis, and powdery mildew is an airborne disease caused by Podosphaera xanthii. And Aphis gossypii is vector of many viruses, and heavy colonization can result in plant death. So the consequences of these biotic stress can be severe and result in significant yield losses and food quality depreciation. So genetic resistance is a preferred control strategy since it helps to reduce the large number or large amount of chemicals used in melon crop production and being friendly with the environment and human health. The development of biotic stress-resistant plants is an important objective of plant breeding strategy, and the development of resistant cultivars that produce high yield but also with excellent quality is challenging for breeders. Normally, breeding for resistance includes several steps. First of all, we had to do the screening of the germplasm collection in order to identify sources of resistance to the pathogen or to the pest in question. And this step is a crucial step in breeding program because the accurate phenotype must be selected in that step. And if we don't phenotype it properly, we'll never find the appropriate gene or the candidate gene and the molecular markers related to them. Secondly, we have to study the mode of inheritance by making crosses between resistant and susceptible accession in order to identify if the resistance is monogenic or oligogenic or polygenic, if it is dominant or recessive. And we need to know this in order to choose later the appropriate methods to introduce this resistance. Thirdly, we have to introduce the resistance traits into elite cultivars. And here we can use conventional methods or the biotechnological approach. For the conventional methods, So depending on the gene of resistance that we identified, there are several methods and we can choose one of them. If only we had a major gene to transfer, so we can do backcrossing. If not, we can do pedigree selection, and there is other ones. The simply inherited genes are easier to transfer, and more genes are implicated in the resistance, more complicated gonna be the method that we will use. And we have hopefully the new biotechnological approach that are based on molecular assisted selection, for example, also genome sequencing, association mapping. All these are approach more precise and useful for breeding program. So in this presentation, we will focus on the first step, the screening, and how to select the desired phenotype using phenotyping and molecular approach. The screening of germplasm is a very crucial step in breeding program in order to select the accurate phenotype. Plant breeders or farmers were making selection based on phenotypes long before the discovery of the DNA and molecular markers. But the classical methods that are used largely for the phenotypic characterization of disease resistance are visual scoring by experts, which is time consuming and can generate bias between different experts and different experimental repeats. So in the phenotyping, the expression of the disease is affected by the virulence of the pathogen in one hand and the background of the plant in the other, and the environmental factors, temperature, light, and so on. And for the phenotype, it refers to the set of observable characteristics resulting from the genotype and its interaction with the environment. And we have to incorporate for the quantitative traits environmental data in the analysis in order to come up with the accurate phenotype more reliably. So establishing the connection between genotype and phenotype is very important. So phenotyping as important as genotyping in establishing the relationship between genes and traits. So in order to be more precise in selecting the resistant plants, a molecular screening needs to be done if linked markers are available, obviously. And these molecular markers analysis will allow a rapid and more precise screening of plants in breeding program. In melon, several resistant genes to Fusarium melonis and to the Aphid have been genetically characterized. We have three major genes, FOM1, FOM2, and VAT gene. FOM1 and FOM2 confer resistance to Fusarium melonis races 0, 2, and 0 and 1 respectively. And we had another matter gene, VAT gene, this amazing gene that confers both resistance to the aphid colonization and also the resistance to the virus transmitted by this aphid. For powdery mildew, several resistant genes were reported and several QTLs were also reported, but most of them are race specific. So the objective of this research is to identify melon landraces resistant to main fungal diseases, Fusarium and Powdery, and to Aphid also, using phenotyping and molecular marker approach. To identify melon accession with multiple resistance for melon breeding, but above all this is to obtain a detailed phenotyping characterization of landraces for breeding and conservation strategies. Before starting a breeding program, we need to know the races of the pathogen. Fusarium melonis and the Podosphaera xanthii had several races. So we have to identify which race is in the area in order to make the appropriate screening and the appropriate breeding program. So identifying the races of the pathogen would aid in cultivar selection for disease management. And this race pathogens identification is based on melon differential cultivars. In the case of Fusarium melonis, 4 races are described worldwide. We had the race 0, 1, 2, and race 1.2. And in order to separate between the races, we use 3 differential cultivars that carry the gene of resistance or not. And depending on the interaction between the gene of resistance and the race of the pathogen, we can identify the race in question. Here in Tunisia, the 4 races of Fusarium melonis were identified in Tunisia with predominance of the race 1.2. This race is the most virulent and the most harmful race. For Podosphaera xanthii, 28 races or more of Podosphaera xanthii have been identified according to their reaction on different melon knives that are used by Lebeda. But the most extended races in the Mediterranean basin are races 1, 2, 5, and 3.5. And these are the cultivars used in order to identify the different races. Here in Tunisia, it seems that race 2 is the most predominant, but we need to confirm the results. So for the screening for resistance to Fusarium wilt, in general for doing screening, we use artificial inoculation. And here the methodology used for the artificial inoculation is to prune a little bit the roots of the seedlings, then dipping them in the conidial suspension of the different races of pathogen. Seedlings were then transferred into plastic pots and maintained in controlled growth chamber at 28-20 degrees Celsius and 16 hours of the period. Plants were maintained during 1 month and symptoms were evaluated. Basic on a scale from 0 to 4. This scale is used to evaluate periodically Fusarium symptoms. And these symptoms, the typical symptoms of Fusarium is yellowing or wilting, depending on the race used. And this scale 0, it seems no symptoms for that plant. And 1, 2, 3, it depends on the percentage of the leaves showing the symptoms. So scoring in different scales helps in making the categories or classes of phenotypes. It means that we can classify if the plant is immune, if it is highly or moderately resistant, if it's highly or moderately susceptible, in order to choose later the resistant genotypes. Also, in order to confirm the foliar symptoms, we cut the vascular tissue in order to observe if there is necrosis or not, in order to confirm the response as resistant or susceptible. The results of this screening is shown here in the table, and we observe that several landraces show the resistance to race 0, 1, and 2. This resistance to race 0, 1, and 2 is mostly qualitative. We observed resistance or susceptibility. There were no intermediate classes. And this resistance is controlled, as we said before, with 2 mature genes from 1 and from 2. For the resistance to from race 1.2, it's mostly quantitative. It is polygenically inherited by several genes. And here we had the distribution of melon accession upon the inoculation by race 1-2, according to their AUDPC values. The AUDPC is the area under the disease progress curve. This parameter, it integrates both the intensity of symptoms and the time taken between inoculation and expression of symptoms. So it is important to give the quantitative and DPIS rather than qualitative scores. It's always better to score on a normal curve rather than discrete, especially for the quantitative traits. And here we observed a large variability of response to race 1.2. There are partial resistance. We obtained moderately resistant, highly resistant, and highly susceptible accessions. And the most interesting thing is we had five accessions with high level of resistance. And the resistance in these accessions must be studied in order to confirm how many genes are implicated in such resistance. We also did the screening for resistance to powdery mildew. This evaluation was done under natural infection in greenhouse condition. And here also we evaluated the symptoms. The typical symptoms are white colonies on deep surface, white powder, consisting of mycelium and spores of the fungal pathogen. And the scale used is 0 to 4, depending on the leaves attacked by the fungus. And also we calculated the disease index in order to evaluate the symptoms. The results show for the disease incidence and the distribution of the melon landraces according to leaf area covered by the pathogen. The results showed large variability of response to powdery mildew from resistance to susceptibility. For accessions that are presented in pink color were highly resistant. They showed disease incidence not exceeding 10%. And also we had 11 accessions presented here in blue color that are moderately resistant, not exceeding 25% of the disease incidence. So a complete characterization of the resistance is necessary by making crosses between resistance and susceptibility in order to see if it is controlled by several genes or digenic or monogenic. The most interesting of these new sources of resistance is that the resistance to different races of powdery mildew has been generally found in accession from India, and the sources of resistance are quite different from commercial type, which make it difficult to eliminate the undesirable melon characteristics integrated into the commercial melon lines together with the resistance genes. So here, these new sources of resistance have agronomic traits and fruit characteristics similar to commercial types of melon, and this is very interesting in breeding program. For the screening for resistance to the aphid, here the evaluation was also done under greenhouse condition, under natural infection, and we evaluated the leaf curling, if the leaf curled or not, and the colonization, if it's heavy or moderate, or there is only few aphids in the leaves. And as shown in the table, large variability of response to aphid was observed, and 9 accessions did not show any curling symptoms and are considered as tolerant, and they may contain the vagine that is responsible of the resistance of the aphid, but we need to confirm this doing molecular markers in order to confirm the presence of the vagine in this accession. The most important thing of this strategy is that 4 accessions were multi-resistant. They showed resistance to the powdery mildew, but also to the aphids. Another independent experiment was also done in lab condition in Spain, and here we evaluated 14 accessions Among them, only one accession showed resistance to the Aphis gossypii. They showed antixenosis and antibiosis, controlled both of them by the bad gene. In the first case, we observed that we have Aphid disturbance behavior, which means that the aphids tend to leave the resistant plants. And we had also aphid disturbance fecundity, what means that the number of nuts produced by aphid was lower than on susceptible plants. It is important to increase understanding about plant reaction to different stresses and in which way genotypes differ in such responses. The use of molecular markers, if available, obviously, is of great interest because the selection of resistant genotypes can be greatly improved by integrating molecular markers with the phenotypic screening. For the three major genes that we showed before, tightly linked markers have been reported. And we have used in our experiment the developed linked markers for F12 and F11. These linked markers are reported by our group team Muru Deal. And for the bad gene, use the linked markers developed by the French team Doggy-Manuel. The results of the molecular screening showed that for the molecular markers linked to FOM1, allowed the detection of 7 homozygous genotypes and 6 heterozygous genotypes for FOM1 gene. And the molecular markers linked to FOM2 allowed the detection of 2 heterozygous genotypes. The screening of the 14 accession showed the presence of that gene only in one accession. So as melon breeder, we have to know more about the landraces, not only the resistance that contain, but also its yield, fruit weight, its sugar content, shelf life. An agromorphological characterization is needed in order to make the appropriate selection of the phenotype. And also, a well-characterized crop genetic resource is a precondition for effective breeding and genetic conservation. Many phenotypic traits were evaluated related to the flower, to the fruit, and to the seed. And these traits were selected from the descriptor list of above. Here are mentioned in the table with all the details that we evaluated. And as shown in the picture, we observed a large diversity of the melon landraces. So we did a phenotypic clustering in order to separate the different melon genotypes. And here we observe there is two groups, the sweet and non-sweet groups, but also there was differentiation between the different genotypes depending on the fruit shape, the fruit weight, and other distinctive traits that permit the separation between the different genotypes. So the evaluated melons expressed a wide range on phenotypes for morphological fruit characteristics, taste, and composition, as shown here in the dendrogram. However, the expression of most of the quantitative traits, especially for the fruit weight and fruit number, the yield, are generally influenced by environmental factors and cultivation practice. That's why incorporating environmental data in the analysis is crucial. You need to do the experiments in different locations during various years in order to make the appropriate selection of the phenotype. Also, a phenotypic correlation was studied in order to determine which ones are more related. These traits relationship are important to the early selection of plants or to the simultaneous selection when more than one trait is desired. So the knowledge of phenotypic variation and traits relationship will assist crop breeders to develop the most adaptive and productive cultivars. So for a more precise clustering, here we used several molecular markers, and to be more precise, we used SSR markers, microsatellite markers, in order for a more accurate clustering. Indeed, integrating molecular markers with phenotypic data allowed a more precise clustering. So it is, I insist here, because it is important to integrate molecular markers in the phenotypic evaluation. So phenotypic tools in common use, or what we call classical phenotyping, are largely labor intensive. They are based on manual measurement of phenotypic traits. You have to measure, you have to go to the field, take measurement for the symptoms of the disease. We have to make visual evaluation, scales, disease index. So it's time consuming, also costly, and most of the time require distraction of plants at fixed time to determine specific traits or evaluations. So high-throughput phenotyping platforms that have been deployed in growth chambers and greenhouses will help raise precision, reduce labor through mechanization, improve data integration and experimental design. There is no need to distract plants. So this technology is needed really. However, these platforms are designed for a limited range of species. For example, in the case of melon, recently, only in this few years ago, that this high-throughput phenotyping just started to be done. So for case of melon, so there is the fluorescence imaging for powdery mildew and X-ray imaging and digital processing for melon seed quality. These platforms also are not available for all the countries, so we cannot all access to this platform, and we still do the phenotyping in classical phenotyping. In order to enhance the classical phenotyping, we do a little bit molecular markers in order to be more precise in our selections. So as concluding remark, we showed here the importance of the local landraces as a valuable genetic reservoir and the need to plan strategy for its conservation and utilization in breeding program. There is a need, an urgent need to preserve the genetic resources threatened by genetic or erosion. We obtained here, we identified more precisely resistant accession for fungal diseases and for Aphid. Multi-resistant landraces were also identified, and this can be used directly by local farmers or as sources of resistance in melon breeding program. For breeding, there is a need of accurate phenotyping. So if it's not done properly, the phenotyping will never find the candidate gene that we are looking for. So integrating molecular markers with the phenotypic screening will greatly improve the selection. So further phenotyping to abiotic stress, drought, and salinity tolerance of these landraces or of other collection is needed in order to develop varieties able to cope with climate change challenges. For this phenotyping to abiotic stress, we can high-throughput phenotyping can be used. Normally, it's used frequently in the European country. And for such abiotic stress and to study the root architecture, collaboration, I think, is needed in this topic in order to make a precise phenotyping to the abiotic stress in melon landraces. I'm really grateful to my colleagues in Tita Zaragoza, Spain, and La Mayora, Malaga, Spain, for the molecular markers analysis done in their laboratories. I'm really grateful to their collaborations and for some experiments done also in the laboratory in order to find to do the screening for Aphid and Ovidur. Really thankful to them. Thank you for watching the webinar. Hope you enjoyed. And here is my contact. And looking forward to see you in the Q&A session that we have 8th of July. Thank you again. Thank you.